We have with us today Dr. Kerry Kinney. She is a professor in the Department of Civil, Architectural, and Environmental Engineering at the University of Texas at Austin and in the Department of Population Health at the Dell Medical School. Her research group examines exposures to environmental pollutants in the built environment and develops technologies to mitigate these exposures. Most recently, she's been working with an interdisciplinary team across campus on whole communities, whole health. And I want to thank our co-chair, HUD, for suggesting this wonderful topic to us today, and she's going to speak to us on a filter forensics approach for exploring environmental exposures and asthma triggers in homes. And I think it's going to be very interesting. Kerry?
Thank you very much. So thanks very much for this opportunity to share some of the results of our filter forensics investigation for essentially looking at whether we can use the dust that we recover from home heating, ventilation, and air conditioning filters to explore environmental exposures and asthma triggers in residential homes. As you mentioned, this is an interdisciplinary and a collaborative effort from many folks across the University of Texas at Austin campus, and also Jeff Siegel from the University of Toronto. I think both Sharon Horner from UT Austin and Jeff Siegel, I think, will be on the line as well. So if we have questions at the end of my talk, we can certainly ask them as well. The motivation for this work is what, of course, this group knows a lot about it, which is that we spend the majority of our time indoors, and much of that time is spent in our homes, where we have complex exposures to microorganisms, chemicals, and allergens. And it's actually pretty challenging to assess these exposures and what the associated health outcomes are associated with those exposures. So what occurred to us is that what if we can use what's already in our homes, or in many homes, as an integrated sampler of the indoor environment? So that's where we came up with this concept of filter forensics, which is to use the heating, ventilation, and air conditioning, HVAC filters, as long-term airborne particle samplers in homes. These are widely available. About 70% or more of residential homes in the U.S. have central forced air HVAC systems, and these come with these filters built in. These filters are easily collected, and they provide an integrated sample of the indoor environment in the homes. The other motivation, of course, is what you guys have been discussing already, is childhood asthma. It's the leading cause of childhood disability in the U.S. There's been a lot of environmental exposures associated with either the development of asthma or the severity of asthma. These include, of course, mold, bacteria, moisture, and a whole range of allergens, as well as certain classes of chemicals. But we often view these exposures as only negative, but of course there's recently been a lot of work looking at protective factors. So for instance, you've heard the term farm-like microbiome. In other words, can microbial exposures protect against the development of asthma and other diseases? And so we're certainly interested in determining, of course, triggers for asthma in homes, but also if there are protective factors that we can encourage. So if you look at microbial exposures, some of the original work in this area have looked at, what you see there on the Petri dish, right? We do culture-based studies, which gave us good insight into the viable or culturable communities that are present. But with the advent of high throughput sequencing that we can do via, essentially extracting the DNA from dust samples and other media, we can get a much richer idea of the range of both bacteria and fungi present in homes. So if you look at that multicolored ball there, each of the different colors represents a different taxa, for instance. So it's a very rich and diverse communities present in homes. The sources for these communities, the microbiome, or indoor microbiome as we call it, are quite complex. There are both indoor and outdoor sources. Certainly people, for instance, the skin microbiome is an important contributor to the indoor microbiome, at least with respect to bacteria. Another area of interest are these semi-volatile organic compounds, SVOCs. These are ubiquitous in the indoor environment. We use them both in the materials that we use to build our houses as well as the materials we use to furnish our houses. Biomonitoring data indicates that over 95% of the U.S. population has been exposed to a wide range of these SVOCs. They're commonly used as plasticizers and also as flame retardants. The content in different products can range from, a few percent to even tens of percentages. So they can be very concentrated sources in homes. And there's a range of adverse health effects that people are investigating. Dr. Zhu, who's working with us on this project, has examined some that are potentially carcinogenic, may be responsible to decrease fetal growth. Some folks are looking at it to see whether it's related to autism, hormone disruption, and altered behavior at early stages in life. So what we decided to do is see what can we learn from our home filters. So this is a filter forensics study, and it was in a sample of rural, semi-rural homes. We had 40 homes that had children with asthma, another 20 that didn't, control homes essentially with no asthma. We put a new filter in the HVAC system, left it there for a month, came back and collected the filter, and then we examined the dust that we recovered from that filter for a whole range of analytes that you see there listed on the right-hand side, so fungi, bacteria, selected allergens, SVOCs. We also characterized the home environment and the air conditioning system as well. As you can see in this study design, we essentially are looking at 60 homes sampled twice, once in the winter and once in the summer. So the dust recovered from the HVAC filters, we used Illumina, high-throughput sequencing, MySeq, to examine the bacterial and fungal communities that were present. We also looked at the chemicals released indoors, specifically selected plasticizer and flame retardants using GC techniques. We also examined fungi, moisture-associated fungi, microbial loads, and other allergens that are listed there at the bottom. I'm going to focus this talk on the first two categories there. So one of the first things we wanted to do was actually characterize how long does an HVAC system run in these sample of homes. And working with Dr. Siegel at the University of Toronto, we came up with a pretty low-cost way of getting run times for the HVAC system, which is essentially how long your fan is actually operating. You're actually in either cooling or heating mode. So we put these monitors, you see the onset monitor there, we mounted it in the supply registers. And what it did is every time, for instance, the heat would kick on, the temperature of the air blowing across that sensor would suddenly spike, and that's what you see there on the figure on the left-hand side. So you see the spike in the black trace, which is the actual temperature that was recorded. And then similarly, if it was cooling, the temperature would rapidly drop. This is kind of an interesting trace because you have both heating and cooling in the same day, and that indeed was one of the houses that we were looking at in the Central Texas area. This is actually possible in our world. But anyway, you can see we developed an algorithm then to come up with the run time. Often the system was either heating or cooling. We also measured the airflow rate going across the filter and the filter efficiency. And the idea was to go from this filter forensics concept, which is essentially recover the dust off the HVAC filter and analyze it for the concentration of different contaminants, to get at, can we estimate, if we know the flow rate of the air that's gone through the filter and the runtime and the efficiency, can we estimate the airborne concentration of these different contaminants? And so that's what we refer to as quantitative filter forensics. If we look at the microbial analyses, essentially we would recover the dust off of the filter via vacuum technique, do DNA extraction via Illumina, sequencing, and then, for instance, for the bacterial communities, we'd use the CHIME bioinformatics pipeline and R and other things for the statistical analysis. So for the SVOC analyses, it was a somewhat similar process in terms of vacuuming the dust off the filters. However, to minimize any cross-contamination with any plastic containing or plasticizers, there was a special aluminum nozzle that was developed to minimize any phthalate or organophosphate contamination of the samples. They were then extracted and run through the GC. We had 6 phthalates, plasticizer compounds, and 5 organophosphates compounds that we targeted in this analysis. In terms of our participants for this particular study, we had a total of 58 school-aged children, of which 2/3 approximately had doctor-diagnosed asthma. Approximately half of those had been involved in an education program, and the other half had not. And then we had another 19, 20 children who served as controls who did not have asthma. 35 boys, 23 girls, average age about 10. It was a predominantly Hispanic population. That's reflected in the numbers you see there. In terms of the types of homes, we had about, 39 homes, detached single-family homes. We also had 19 families that were in manufactured homes. You'll also hear the word trailers. So we had a split in the different types of homes. Looking at the socioeconomic status of the participants, you can see that they were on the lower end of the SES spectrum, particularly those that were living in the manufactured or trailer homes. We also gauged the severity of asthma for the children in the homes. This is based on parent ratings of the frequency of symptoms, including their daytime asthma symptoms, nighttime awakening, days with activity limitations. Based on this classification scheme of the children with asthma, 24 had intermittent asthma and 16 had persistent asthma. So let's start with some of our results. This is looking at the run time, so how essentially long the HVAC system was running during our 1-month sampling time. We looked at both detached and manufactured homes because, again, we had 2 different types of homes, and we found that they both had similar AC usage. So that's the graph you see in the top left there. However, we did indeed prove that Texas is a hot and humid environment, and so the summer run times for the HVAC systems were substantially larger than those in the winter. And this is the data. These runtime data is what we used, and I'll show you in a moment, to essentially convert our dust concentration into estimated airborne concentrations. If we look at the bacteria that are present in the HVAC filter dust, this is looking at the genus level in a sample of about, I think it was about 20 of our filter samples. And again, what this figure represents is each different color is a different taxa, and the greater the abundance of a specific taxa, the larger the circle, essentially. And what you can see is there's a significant human imprint evident in the HVAC filter dust. So the taxa there that are underlined are those that are typically associated with humans, and specifically the skin microbiome. And in fact, if you dig in a little bit more, this is looking at a single filter and looking at the percentage of, or essentially the relative abundance of human-associated bacteria, and specifically those associated with the microbiome on the human skin, and you'll see it's roughly about 5%. You'll see that we've taken this particular figure and we've divided it up into cut, swab, and vacuum, and what that reflects is we did some earlier work to try to figure out different ways of recovering the dust off the the dust off the HVAC filter, and see what effect it had on our microbiome results. And based on those analyses, we decided that the vacuum gave us the most consistent results. So that was the technique we used to recover the dust off the filter for these analyses. So the skin is perhaps a little bit more palatable, but we also have a pretty recognizable imprint, if you will, with stool-associated taxa, and that's what's shown here. Again, it's anywhere from, depending on the method, but roughly 5% of the bacteria recovered from the filters was associated with fecal matter. Now, if we transition, if you will, over to fungal diversity, again, we're showing a similar representation of the different fungal taxa that are recovered from the dust on these HVAC filters. And what's interesting here is that these are all very common taxa, normally found in the built environment. Many of these, for instance, the alternaria, are common outdoor taxa that make their way indoors. There's actually a pretty low percentage of human-related fungal taxa in these samples. So who else is there? Well, not only do we have heat in Central Texas, we also have cockroaches. And it was interesting that we found Bladobacterium species, which is an obligate bacteria found in the cockroach gut. We found that homes that reported cockroach problems had higher levels of this Bladobacterium in the HVAC filter dust than those that didn't report problems. And if you're wondering if your pets contribute as well, we did find, for instance, if you have an indoor pet, and in particular this is looking at the dog, we could detect the dog allergens in the HVAC filter dust as well. So we were curious if we could see if there's a relationship, in this case, between the bacteria present in the homes and the asthma for children in the homes. And so what we're showing here are 3 categories: homes with no asthma, homes with children with intermittent asthma, and homes with children with persistent asthma. And we have 4 families there listed on the right that had been shown previously to be protective against asthma development if children were exposed to these taxa in these families during their first year of life. It was found to be protective against the development of asthma, and we were curious what it would do in terms of our asthma classification. And what we did find is that the homes of children without asthma in this study had higher concentrations of selected types of bacteria than the homes of children with intermittent or persistent asthma. In other words, this idea that is being explored actually in many different studies of a protective microbiome appears to be at play here in this set of homes. And if you drill down a little further, we find, and you go to the genus level here, we find, and you look at stool-associated bacteria and asthma, again, we see that non-asthmatic children in this study had higher abundances of stool-associated taxa in their homes than children with intermittent or persistent asthma. And so, again, this is supporting a growing body of evidence that a protective microbiome may indeed be possible in homes. Now let's transition to fungi. And what we found is, of course, it's a mixed bag. So some fungi were present in higher abundances in homes with asthma, and others are found in higher abundance in homes without asthma. So homes without asthma, for instance, shown in red here, had higher abundances, if you will, of Saccharomyces, which is actually the presence of high concentration of yeast has been shown in other studies to be perhaps protective against asthma development. And then if you look at those fungi that appear to be enriched in homes with asthma, the yes, in other words, the green there, there's several taxa there that have been found in other studies to also contribute to allergic respiratory responses. So there is some consistency there. When we just pulled out, potentially allergenic fungi and looked at their grouping relative to whether they were with asthma, so those are the 2 groupings on the left, one with the educational ACT intervention, one without, versus the control, the no asthma case, we didn't see a statistically significant difference in this case. Then we started to dig a little deeper, and we started to examine the relationships between the diversity of fungi that are present in homes and home factors, SVOC concentration, and asthma status for the children in the homes. So we have 2 columns there. One is richness overall, means across different seasons, and that just is looking at the number of species, unique species that are present, whereas the Shannon index, which is the next column, combines both the number of species that are present and also their relative abundance. So it's a measure that considers both of those factors. And we have 2 values there, one for the p-value, and also looking at the FDR is looking at the case when we consider a more stringent case where we're looking at multiple comparisons. So we're providing a bit more stringency on the data. But bottom line is if you look at, and the ones that are statistically significant are bolded there, and this is just a subset that we looked at, but we find that there's a relationship between the age of the home and the diversity of the fungi present in the samples, the AC usage, and also, interestingly enough, phthalates, or specific phthalates and flame retardants that are shown there in the left-hand column also seem to be related to the richness and diversity of the samples. Now, if we look at which variables were associated with differences in fungal community structure, so the overall fungal community, we found that socioeconomic factors seem to affect it, proximity to agricultural fields. These were rural, semi-rural homes, and if they were within 1,000 feet of a field that was under cultivation, we included that. We also looked at the fraction of carpet, air conditioning use, again, specific phthalates and flame retardants, moisture-associated fungal concentrations, and finally, asthma. All of those were factors that seem to affect the overall structure, in other words, membership, if you will, of these communities, these fungal communities present in the filter dust. If we dig in a little bit deeper and we look at there's kind of two ways to visualize this, the differences in fungal community structure as a function of several parameters. So, for instance, season has been shown previously to greatly affect the airborne fungal community, and indeed we see that reflection in the filter dust communities as well. We also, again, found proximity to an ag field under cultivation to be an important parameter. The severity of asthma, so whether, in this case, we define that as whether it's intermittent or persistent asthma, or no asthma, and then finally by asthma group, in other words, whether they've had the educational intervention, asthma, no asthma. Now we can transition off of the microbial exposures to look at the SVOC, semi-volatile organic compound levels in both settled and HVAC filter dust for these samples. So the blue circles, if you will, are the settled dust samples. The red is the filter dust, and then the grayed out, triangles there are values collected from 42 studies representing about 4,000 homes. And you can see in general that the SVOC levels that we see in the settled and HVAC filter dust are on the same par as what's reported in the literature. The most frequently detected phthalates was DEHP for both HVAC, filter, and settled dust. And the compounds that were detected at the highest concentration in both filter and settled dust in our sample were DEHP and BBZP. These compounds are widely used as plasticizers in PVC products, in other words, flooring materials and building materials as well. And in some cases can be up to 10 to 30% by weight, these materials. If we look then a little more deeply at DEHP and BBZP specifically as a function of the different flooring materials, we see indeed that the red bars there are homes with PVC flooring. Another term you'll see is vinyl, so vinyl flooring. And those without, you'll see that there are significantly higher levels of both DEHP and BBZP in homes that have the PVC flooring. What's also interesting is if you look at that's in the top graph. If you look at this graph below that, if you take out then homes that have the PVC flooring and then just compare those homes with and without carpet, we also see that DEHP and TDCPP levels in settled dust were associated with the presence of carpets when homes with PVC floorings were excluded from the data set. So in other words, this is just showing, these are in settled dust samples, it's just showing the importance of the different materials that are used in homes and what that means with respect to the SVOC concentrations, in this case on settled dust. We also saw in that data set we were curious about exploring the association between DEHP levels and asthma. And what we found is if there was in homes with kids with no asthma, we didn't see any type of relationship. But in homes where the children did have asthma, there was the DHP concentrations in the filter dust of the children with asthma positively correlated with asthma severity for samples collected in the summer. The relationship between DHP exposure and asthma is still a very active area investigation, so just correlation does not mean causation in this case. What's interesting, though, is we're starting to understand there might be a chemical and biological interaction that's occurring. So DHP is degraded by selected fungi that indeed can become enriched in high DEHP environments. So then the question becomes, when you start to look at the role of DEHP in, in asthma, for instance, is it the chemical itself, or is it the fungi that degrade that chemical, which then may have some additional health effects? So that's a very interesting concept that we're going to be pursuing further. So as I mentioned earlier, we wanted to look at not just the concentration of the contaminants and the microbial communities in the dust itself, but also try to use our the information that we gathered on the run times for the HVAC systems and the volumetric flow rates through those systems, et cetera, to quantitatively estimate the airborne concentrations of these contaminants in the home. And I'm showing one set of results here. This is looking at the concentration of total bacteria in cell equivalents per cubic meter, so the airborne concentration of total bacteria. On the left-hand side, we have summer. On the right-hand side, we have winter. And it's just showing the distribution, if you will, in the different samples that we collected. And what we found is, this is one example, but there were several others, of course. There's a factor of 100 times to 1,000 times variation between homes for most of the analytes. So the question was then, can we use ordinary filter forensics? In other words, just the dust concentrations. And what we're showing here is essentially on the x-axis, we have the dust concentrations of total bacteria cell equivalents, and that's the filter forensics component. And on the y-axis, we have the concentration of estimated airborne concentration of bacteria, the quantitative filter forensics. And what we found is that the filter forensics and the quantitative filter forensics are strongly correlated in the study homes. And since HVAC characterization is time-consuming and not always possible, it is reassuring, at least for this sample of homes, that the filter forensics appears to be sufficient. Okay. Of course, if you had disparate types of homes and multiple climates, et cetera, certainly that would need to be examined more carefully. And with that, I just want to summarize some of our findings on the filter forensics. Filter forensics can provide valuable insights into home environmental exposures. It's nice to be able to use what's already available in homes. We found diverse bacterial and fungal communities recovered from the dust in these homes, and they include a strong human-associated signature, which simply reflects that these are occupied environments. In our study, the homes of children without asthma had higher concentration of selected types of bacteria than the homes of children with intermittent or persistent asthma. Selected fungi we found were enriched in homes with asthma, and then others were enriched of homes without asthma. So this gives us some clues on things that we can follow up on. All of this, though, is implying that there are both potentially beneficial and potentially detrimental microbes present in residential homes. When we looked at the SVOC, specifically the phthalates and the flame retardant concentrations, we found that in general they correlated well with what we found in the filter with those in settled dust. The DHP concentrations may be associated with asthma severity in homes of children with asthma. Certainly that'll need a lot more study, but again, it's kind of feeding into this literature into studies that now folks are focusing on DHP. We found filter forensics allows for a comparison of exposure in similar homes. And quantitative filter forensic directly estimates the concentrations, the airborne concentrations and exposures, so that can be useful. And of course, additional studies with a larger number of homes across several climate zones will be useful to verify and extend these findings. Kind of what is the future? One thing that we would like to certainly improve is to see how we can engage with communities in both the collection of the data and feeding that data back to our communities. I'm part of a Whole Communities Whole Health initiative here at UT Austin. That is gearing up to do just that. Specifically with regard to the filter forensics, we couldn't help ourselves, so we have filter forensics, quantitative filter forensics, and now we've termed, of course, community filter forensics. And the idea is, can we couple a mail in your filter dust campaign with self-reported home environment, child asthma status data via smartphones, for instance, to more efficiently gather data from a far greater number of homes. And then, of course, we would do similar types of analyses that we've done before, which is the microbiome, SVOCs, and allergens, for instance. We're also interested, and this is what we're doing actually as part of our new HUD study, is to look at the pro-inflammatory potential of the dust. We're working with Dr. Felix Rivera Mariani at Larkin University for that aspect. So our plan going forward is that we have two phases. One is to compare in an urban cohort the in-person data collection method that we used in the HUD study I just described, and then compare the results that we get for that with the community filter forensics approach to see how to validate the method and see how they compare. And then the next phase is to take this community filter forensics, once we've fine-tuned it, and roll it out to 3 different cities, in this case across Texas. And the one advantage of Texas being so big is that it has 3 different climate zones. And so we'll be looking at Houston, Dallas, and Amarillo. And we've also added some more folks onto our team, Dr. Matsui for the asthma, Felix, who I've already mentioned, for the pro-inflammatory potential, and other folks for the phone app and real-time monitoring applications. And with that, I'd like to go to the final slide here, which is just a set of pictures, I believe, of the folks that were involved with the HUD study that I just described, the original HUD study. And again, these are folks at the University of Texas, and then Sharon Horner's in the top left, and Jeff Siegel's second from the top on the left. Hopefully they're also joined the call as well. And with that, thank you for your time.
Thank you so much. I was right. I was as interested from just looking at the slides themselves as I was hearing you actually walk me through them. Lindsay, do we have any questions?
Yes, Lorene, we have 3 questions from the audience. I'd like to just remind listeners for the last time, if you have any questions that you would like to ask Dr. Kinney, please use the questions function on your GoToWebinar panel. So the first question is, were homes consistent in terms of carpet versus hardwood floors in regards to the taxa found or quantities of taxa found, specifically filter bacteria and fungi?
Filter bacteria and fungi as a function of carpet. Well, it depends on whether you're talking the composition of the, sorry, can you repeat the question?
Sure. I think the question was just asking, were the findings consistent or were homes consistent if they had carpet versus hardwood floors, specifically for those bacteria and fungi?
In terms of the results that we saw. We did have, if you look at, let's see, the differences in the community structure, like who's present, like the whole community, we did find, for instance, the fraction of carpet that was present in the home did have an effect on the taxa that were present. I don't think we saw it with the hardwood floors. I'd have to go back into the data set. So that was specifically for the fungal community structure. I was trying to come up with the number. I think it was 40%. I'm trying to remember. About 40% had carpet, I believe, and the rest were some sort of mixture of other things. So hopefully that answers it to some degree, but I don't think we saw a very strong signal other than essentially how much of the house was carpeted did have an effect on the fungal community structure, at least is what we've looked at so far.
Thank you. Another question is, what are the key takeaways from your research that you would share with homeowners? Based on your work, are there any actions you would recommend homeowners take?
Yeah, so I think it does tie to, well, it's interesting. It depends on which aspects you're looking at, right? So I was very interested actually in the previous talk in terms of what was being recommended in terms of minimizing for instance, moisture damage in homes, for minimizing excess fungal growth. I think that still very much stands. What's interesting to me is, and this is something that I can't say that anybody has come up with a very concrete answer to, is that can we have a healthy microbiome that we can dust houses with, right? We are nowhere near that, so just to be clear. However, it is very interesting that we see this kind of protective taxa that other people, like in Finland and elsewhere, have also found to be protective against the development of allergies. So I guess one of the things that I would recommend is that, all microbes are bad story is not the truth. On the other hand, certainly getting rid of pests, excessive moisture so you don't have, fungal damage, because I think that exposure is a negative. Also, and this is a tougher one, is the SVOCs that we bring into our homes. I started, looking at the PVC flooring, and I think there's a lot of work that needs to be done, certainly before we can make recommendations on that. But given that these are at pretty high concentrations, I think that this should be a very active area of interest. But bottom line, what I tell homeowners, I think, is very similar to what you guys who are doing the asthma intervention studies are doing, is to reduce clutter in the home, cut down on pests, cockroach, basically pest control, hardwood floors, because you can reduce the you can clean them with wet mopping versus dry, like brooms or vacuum cleaners that'll just redistribute the material. Using the vent stove to reduce particulate matter concentrations. Some of the things that I think are kind of standard in the asthma intervention, I don't think those have changed. I think we're just starting to kind of dig in, though, into different things that may be things that we recommend in the future. For instance, maybe we recommend different types of flooring materials and other things. But, of course, that's going to need a lot more research.
Thank you. And our last question before I pass it back to Lorene is, have you applied filter forensics to filters in portable air cleaners?
I have not, although I've thought about it. One of the, that would give you some interesting information that, for instance, if you're using one to clean the child's bedroom air or whatever, that would give you some information, at least more local to that one room in the house. So I would suggest that that's a great idea. One of the concerns I always have with any type of filters is, of course, you don't want to get it to overloaded and moist so that you have a problem with it turning from something that is, in essence, removing some microorganisms, of course, with the dust, to one that could be a source.
Okay, great. Thank you. That's everything on our end. Lorene, if you want to take it from here. Thank you.
Thanks, Lindsay. We have 2 questions in the room. I think it's only 2. Okay. We'll start with Jason.
Yeah, I was interested. You made a remark, and I'm not sure if I heard it correctly. It was about the fungi present, and I heard the word yeast being used. Is yeast something that is connected with this at all?
Well, so if you look at that the Christmas-colored slide, which is green and red, what about the fungi present? And it shows asthma, no asthma.
Right.
There's Saccharomyces, the family anyway that Saccharomyces is in, which is yeast, was found to be enriched in homes with no asthma. And there'd been some studies where, I think it was they'd collect, not our study, a different study where they had collected floor dust on the, from homes of children when they were between, I think, 2 and 3 months old, and then they tracked them over time to see how many, of course, developed asthma, and they found that those homes that were enriched in yeasts had reduced odds of developing asthma by age 13, if I recall correctly. Now, that was a culture-based study, if I'm remembering the right study, but it's interesting because here we are in a culture-independent study, using DNA sequencing, and we find that perhaps this is something that's enriched in the no asthma home. So is it related or not?
Thank you.
Mm-hmm.
The other question is, in your study, filter forensics, did you test the difference between the upstream side of the filter versus the downstream side of the filter? And where I was going with that is, if you're finding this on the upstream side that's capturing all the particulates and microbes and whatever,
mm-hmm.
and you're not seeing that in the downstream, would it be more advantageous from a remediation standpoint to use like a HEPA filter that clears out the materials more so than just some of the other types of filters? Did you do that testing or look at the upstream versus the downstream, and did you analyze for different filter types? Like I say,
So we, to normalize across, and I hope Jeff Siegel's on the line. I'm just going to address the type of filter that we used. If Jeff Siegel's trying to chime in, if you could let him answer. He's our filter expert. But we use the same MERV 7 or 8. Well, we use commercial filters, clean ones, that we put in MERV 7 or 8 rating. So those are not the HEPA ones. So that we would have some consistency across the homes, and we only left them in for a month, which was more than enough for the microbiome analyses and many of the analyses. With regard to some of the allergen analyses, it was hard to actually get enough dust in some cases to do a complete suite of allergen analyses. So, for instance, in our next study, we're doubling that to 2 months. So in our case, we were actually wanting more dust, if you will, on the front end of the filters, whereas many times those filters that I showed you that were heavily loaded, that would have been a pre-existing filter that somebody would have had, in other words, people don't change their filters that often, right? So with respect to that, now if Jeff on the line? If not, I'll take a crack at the HEPA question too.
So 2 comments. Everything Kerry said was right. I would add that something she said earlier in the presentation is really important from the perspective of what filter you pick. One of the hardest part of filter forensics is getting dust off of the filter. The higher efficiency filter you use, the harder it is to get dust off of it. For both some practical reasons, dust gets more embedded in the filter, as well as some kind of logistical issues. Higher efficiency filters, things like HEPA filters, and even high efficiency but not HEPA filters have metal screens and other things for structural purposes in place. And so we have found, we do a lot of filter forensics as well, and we have found that the sweet spot from a filter forensics perspective is something like a MERV 7 or 8, maybe a little bit higher, but in that kind of order of efficiency. So when you start going to higher efficiency filters, sure, you pull more stuff out of the air, but you can't necessarily get it back out from an analysis perspective.
And do you want to comment on you've always talked about bypass. in these situations.
So the other comment about filter efficiency in general, and this applies, by the way, to both HEPA filters, portable filters, as well as installed filters in a central system. There is a huge difference between laboratory tested efficiency and actual efficiency that you get in place. And some of the central issues are things like bypass, where air goes around the filter because the filter is not well seated, to things like short circuiting. If you have a portable air cleaner, it essentially takes the air from the outlet and some fraction of the air goes back in at the inlet. And so it kind of very effectively cleans the air right around the portable air cleaner, but doesn't as well address air farther away in the room from the portable air cleaner. So I think we have to be really cautious when going from the efficiency of a filter, as it's rated on the box or by some standard, to how it's actually going to perform in real life. And in our own research on central filtration in particular, we get that if you go from a kind of mid-efficiency filter to a very high-efficiency filter, in general, you'll get better performance, but nowhere near as good performance as you would expect from the difference in the lab-rated efficiencies.
Yes, and that gets down to, when you recommend HEPA filters, right? So it is better, but it's not as much better as you would expect.
Yeah, absolutely. And, for any type of air cleaning to work, central filtration, portable air cleaning, the context really matters. So there was a question about practical advice to homeowners earlier. And one of the things that I think we as a community need to do a better job about is, the home is a system. And so simply putting in a better filter won't necessarily achieve better results. So homeowners, even though this is a more complex story, it's a more realistic story, addressing issues like air leakage of the building, addressing issues around maintenance of heating and air conditioning systems, addressing issues of things like duct leakage. All those things are part of the picture of achieving the benefits from any type of filtration.
Yeah.
And not to be too recursive, but there was a question earlier about using portable air cleaners for filter forensics. That's something we've done a bunch of here as well, particularly in social housing and low-income housing, where central systems are much less likely to be present. And one of the things we found as a complete side benefit of doing filter forensics with portable air cleaners is that you get a lot of acceptance from potential study participants because they see a benefit of getting an air cleaner in their house. And so we've had very good acceptance of doing filter forensics with portable air cleaners.
Right, probably much more than if you said, I want to go sample your house and you set up a little filter that's just pulling air through a filter. Whereas if you say you can have this cleaner, then, and it might even be more acceptable to folks who are renting homes where the landlord, for instance, may not want you to be testing their central air system, but they don't mind perhaps if you're doing an individualized one. Thanks, Jeff.
Sure.
We actually have two more questions that just came in. Is it okay to move forward and ask them, Lorraine?
Yes, go ahead.
Okay. Was any research done to see if the vent ducts trap different material than the filters in the HVAC system?
Oh, the ducting work itself.
Correct, yes.
Yeah, oh, sorry. No, we did not do that. Of course, that's a pretty interesting area to look at, kind of following along the line of, do you get penetration of material past the filter? And the answer, of course, is yes. One of the questions become, is your duct work essentially then becoming a source? One that's, we have studies that we call hidden spaces, where we look at what I'll call out of sight and generally unmaintained spaces. and and that could be an attic or a crawl space, ceiling plenums, et cetera, that under certain situations we do show that you can get movement of, for instance, microorganisms from those unmaintained spaces into the occupied or room space. But in our particular study, no, we did not look at the ductwork.
Thank you. And then the last question, is it possible that there may be an opportunity in the future to partner with tribes on this type of research?
I would say certainly. I think this is a great opportunity for many different communities because, again, the idea that you could take something that's already present in the home and maybe couple that with, we're learning from this first study what are some of the parameters that appear to be affecting, say, the microbial community, appear to be affecting the asthma status of the kids in the home. And we've certainly refined our questionnaires for the second study that I alluded to incorporate that knowledge. But certainly we're the previous study was more in a rural area. Now we're doing an urban cohort because we want to understand if it changes, some of these relationships that we've seen. And certainly working with tribal communities would definitely be something that we would enjoy doing, and also I think would be of great benefit.
Thanks so much. First, I want to thank everyone on our participants, our speakers, our updaters. Our next meeting will be in October, and then the following one would be February, June, and then October again. So we're hoping to get back on schedule and then add some other ways to interact with people. So with that, I'd say thank you for participating, and we will say goodbye. Thank you all.